So next step of glycolysis is isomerization of glucose 6-phosphate. Isomerization of glucose 6-phosphate is catalyzed by the enzyme phosphoglucose isomerase, also known as phosphohexose isomerase. The reaction is reversible isomerization, and glucose 6-phosphate is isomerized to fructose 6-phosphate. If we look at the reaction, the isomerization of glucose 6-phosphate to fructose 6-phosphate can be visualized as the conversion of an aldose to the ketose isomer. The enzyme is phosphoglucose isomerase. We can clearly see the phosphate group is still present in the 6th carbon, and the carbonyl carbon which is present in the form of 1st carbon is now shifted to the 2nd carbon, leaving the 1st carbon with the hydroxyl group. Now this shift of carbonyl carbon from the 1st to the 2nd, that is the conversion of an aldose to ketose, is significant to understand the further step of glycolysis, which is the phosphorylation of fructose 6-phosphate. So phosphorylation of fructose 6-phosphate is catalyzed by the enzyme phosphofructokinase 1 or PFK1. The reaction is irreversible phosphorylation. Some important features of this reaction are it is a rate limiting step of glycolysis and also a committed step of glycolysis. Now rate limiting step of glycolysis means by controlling this step the whole rate of glycolysis can be controlled. The committed step of glycolysis means once the substrate enters into this reaction, the product which is formed is destined to undergo the subsequent steps of glycolysis. The reaction showing phosphorylation of fructose 6-phosphate. Fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate. Now the phosphate group in the 6th carbon is already present from the first reaction. In the second reaction, carbonyl carbon was shifted from the 1st carbon to the 2nd, while glucose 6-phosphate was isomerized to fructose 6-phosphate. Now in this 1st carbon, another phosphate group is added, and that phosphate group is transferred from ATP to the 1st carbon of fructose 6-phosphate to form fructose 1,6-bisphosphate, and the enzyme is phosphofructokinase 1 or PFK1.